Good morning everyone. So today we are going to learn about class of immunology, which is dealing with antigen and antibody reactions. So before going to start with the class, we should be understanding the learning objectives of this particular topic, which includes principle of antigen and antibody reaction, different type of antigen and antibody reactions, which includes precipitation reaction, agglutination reaction and complement fixation reaction. Here we are going to learn about the precipitation reactions, their principal types, application, advantages and disadvantages. Similarly, we are going to learn about agglutination reactions, principal types, application, advantages and disadvantages, along with complement fixation test, principal, application, advantage and disadvantages. So to start with the topic, what are antigen and what are antibody? You all know antigens are known as foreign substances which are not self for human body. Their composition is generally principal composition of its generally is a protein structure, or it can be glycoprotein or it can be polysaccharide. But these are known to induce immune response in the host body, and they are considered as foreign substances or foreign body by the host immune response. And against these foreign substances, protein material, that could be toxin, that could be certain part of cell wall components of any microbial organism like bacteria, fungi, parasites, and against these components there is always elicited an immune response, and that is in the form of antibodies. So these antibodies, again you all know, antibodies are the response from the immune cells which is against this particular antigen, and specific antibody combines with the specific antibody, and these reactions of specific antigen and specific antibody are known as antigen-antibody reactions, and these reactions are also known as serological reaction, immunological reactions or immunoassays, and these are generally the in vitro immunoassays. Okay, so antigen-antibody reactions, these are biomolecular associations where the antigen is going to combine with its specific antibody in a particular specific way, and that can be also observed in laboratory. Now these tests are done for the purpose of diagnosis of various infectious or communicable diseases. Okay, so what are the stages in brief? The stages of antigen antibody reaction are the three stages. First one is the primary, secondary and tertiary. Okay, so primary stage, this is the initial reaction where the antigen and antibody, they interact with each other. There is an interaction between antigen and antibody, but these particular reaction, when it happens, there is no visible effect of this particular reaction. Again, this is very rapid and irreversible reaction, and it is held by the weak bonds of van der Waals. Okay, follows the primary stage comes the secondary stage, which shows the visible effect, in which we can see the visible effect by the various kind of laboratory tests that includes precipitation, agglutination, lysis of the cells, killings of live antigens, neutralization of a particular toxin, where we can see the Naglus reaction and all. Complement fixation tests are there, phagocytosis is there. Okay, and tertiary stage reactions that are seen in vivo inside the host. So this can be termed as the antigen antibody reaction that are carry out, or they are initiating certain chain reactions. These chain reactions are going to show the effect in vivo, and these effects are either neutralization or destruction of various injurious antigens and during carrying out this particular process there could be damage to the host tissues, and this response can include humoral immune response against these particular infectious agents, diseases, as well as certain clinical energies and other immunological diseases also. So what are the general features of these antigen-antibody reactions? In brief, these are specific reactions, but cross reactions can also be seen due to antigen relatedness, and this you must have studied in antigen, that is the heterophile antigens and all, so that is in that relation. Okay, entire molecule of this particular antigen and antibody, they are going to combine, they are going to react, there is a reaction between them, and they are going to participate in this particular antigen antibody complex formation, and they are going to either agglutinate or they are going to either precipitate. Okay, but there is no denaturation, as we see that antigen antibody complexes combine, and that leads to certain kind of destruction, but this destruction is not at this particular level, this is afterwards. So during this antigen antibody reaction, when antigen combines with the antibody, there is no denaturation of this particular complex, and this complex is always formed on the surface. This combination is always on the surface, and this is firm combination and reversible. Okay, so these reactions are very specific and they can be observable. Hence, extensively these reactions are used as a diagnostic procedures or diagnostic tests for identification or diagnosis of certain infectious agents and diseases in the laboratories nowadays. Now, antigen and antibody reactions are also termed as immunoassays, and most of these immunoassays, they are utilizing serum as a sample, okay, as a specimen for carrying out all these reactions. However, why? Because the serum contains antibodies, that's why. And however, certain samples can also be used like urine or CSF. They can be also used for these immunoassays, because you all know most of the infectious agents, their antigens are released in urine as well as in CSF. For example, cryptococcal antigen is there. Okay. Then diagnostic purpose or diagnostic use of this particular test that includes, these are the immunoassays and can be broadly categorized into antigen detection assays as well as antibody detection assays, where we are detecting the antigen in particular patient's sample using specific antibodies. When in detection of antibody or antibody detection assays, we are using antigen, and these antigens are employed for detection of specific antibody against specific infectious disease in the particular patient specimen. Okay, so we are detecting antibody here using commercially prepared antigen. In the first one, we are detecting antigen using commercially prepared antibodies. Okay, there are two types of assays in antigen antibody reactions. One is the qualitative, another one is the quantitative assay. Qualitative assay that employs undiluted specimen. These undiluted specimen, that means they are containing antibodies. The specimen contains antibodies against the specific antigen or specific infectious disease agent's antigen, and that antibody suspension or specimen is mixed with commercially prepared antigen or vice versa, and they are going to give you a result. This result could be interpreted in the form of either the reaction is positive or either the reaction is negative. Okay, so this is based on presence or absence of either antigen where you are detecting or antibody whatever you are detecting in the particular specimen. Okay, and the exact amount of particular antigen and antibody in the specimen, what you are objective, what you are identifying, is not known or it is not estimated in this particular qualitative assays. Where we are talking about quantitative assays, these are done after we screen the specimen by qualitative assays. Now, these are employed generally in mass screening tests. Okay, qualitative assays where the blood donation camps are there, where you want to screen all the individuals, all the donors with a particular presence of for particular infectious disease. For example, suppose treponema pallidum infection for syphilis. Before blood donation, these all tests are done, and for that VDRL test is done. VDRL can be done in qualitative as well as quantitative. So once the qualitative is positive, like certain amount of antibodies are present and they are mixed with the antigen and they are showing the presence of antibody against the antigen. So this is a qualitative one. But now you want to know what is the significant amount of antibodies are there or not. When we are saying significant amount of antibody, that means in these immunological assays or serological reactions or immune assays, it is said that all of we have a particular amount of antibody against most of the infectious diseases, and this amount of antibody is known as baseline titer. So significant titer is something which is slightly above this baseline titer, which signifies yes, this is because of the infectious agents presence in the host body. Okay, this is called as significant. And now these qualitative and quantitative assays plays most important role. Quantitative assays plays here. Okay, where we are going to know the exact amount of the antibody which is present in the specimen. How we are going to know this exact amount of antibody present in the specimen? Here we are going to dilute the specimen serially, 1:10, 1:20, 1:40. This is how we are serially going to dilute the specimen, and to each of the dilution we are going to add exact amount of antigen. Okay, so exact amount of antigen is going to be added in each of them, and then we are going to see at what highest dilution of the particular specimen, which will be a serum, showing an absorbable reaction, and this is against the specific amount of antigen, and this is called as an antibody titer. So highest dilution of a serum which shows observable reaction with the specific amount of antigen, that is called as an antibody titer, and vice versa we can do it for antigen also. So quantitative tests are more reliable as they can measure the number of antibody as well as they can differentiate between true negative and false negative results. Okay. Now the evaluation of these immune assays depend upon two terminology. One is the sensitivity, another one is the specificity. Okay. Now these are the most important statistical parameters which are applied for each and every immune assay. Okay. Sensitivity can be defined as this is the ability of a particular test which is going to correctly identify the presence of the disease or the true positivity. Okay. So correctly, if the test is positive and the disease is there, then the test has good sensitivity. How this sensitivity is calculated? The sensitivity is calculated as true positive are divided by true positive plus false negatives. Then specificity is another parameter which can be defined as the ability of this particular immunoassay or serological test, which is going to correctly identify those which are not having this particular disease. That means these are true negative. So what we have seen in sensitivity is the test parameter, or it is the statistical parameter which is going to identify true positivity, and specificity is going to identify true negativity. So hence specificity can be calculated as true negative which are divided by true negatives plus false positive. Okay. Now we come towards the next point of the topic is mechanism of precipitation. So here you can see the lattice formation. Lattice is the hypothesis which is proposed by Marek. Here he has proposed three zones of these antigen-antibody reaction. One is called as flow zone, where antigen is excess and antibody is low. As you can see, these are the antigen, this is the antibody, and these are the antigen combining sites over here, where only two antigens can combine these antibodies, while the remaining antigens they do not have exact amount of antibodies to combine, where the zone of equivalence or lattice formation is there. Why? Because there is exact number of antibodies are also available and exact amount of antigen is available, which is going to form this lattice. And this lattice, the lattice is formed, that means the test can be read properly, it can show the results properly, and there is no pro-zone which is antigen excess, or there is no post-zone, that is antibody excess. Okay? So these are also going to explain the similar type of post-zone and zone of equivalence. So serological reactions which we are going to learn today is the precipitation and agglutination, complement fixation, while others are neutralization, immunofluorescence and enzyme immunogenicity, which can be dealt afterwards. So precipitation is what? What is precipitation? Precipitation is nothing but a soluble antigen when it combines with its specific antibody in presence of optimum concentration of electrolyte, NaCl, at suitable temperature, at optimum pH, and when these parameters are optimum, like presence of NaCl, electrolytes, optimum concentration, suitable temperature, that is 37 degrees Celsius or plus minus 2, and then optimum pH, that is 7 plus minus 2, at this particular favorable environment, when these antigen combine with antibody. Now here, see, we are dealing with precipitation. So the first thing is important here is that soluble antigen. So when such things are, favorable things are there, favorable conditions are there, these soluble antigen combine with their specific antibody, they forms a complex. This complex is not soluble. It is an insoluble precipitate. This insoluble precipitate is going to be formed in case of precipitation reaction. And when this insoluble precipitate, instead of settling or sedimenting at the bottom of the reaction, that precipitates, they flow on the surface of that particular solution. This is known as flocculation reaction, or this is known as precipitation flocculation reaction. And the best example of this is VDRL test, okay, or Kahn tube test. So it can take place in liquid media or in gels, or in agar or agarose or polyacrylamide gels. So this is the example of precipitation reaction. Here you can see the soluble antigen is there, antibody solution is there, and both are going to combine and form the equivalence visible precipitate, zone of equivalence and the precipitate. Okay. So clinical application of precipitation reaction, it is the earlier precipitation reactions, where one of the widely used serological tests was the precipitation reactions. But nowadays these tests are obsolete because there are newer, simple and rapid methods are developed, and they have greatly reduced their use. The previously used were various, but nowadays out of that few only includes VDRL test and LXTL precipitation. However, VDRL LXTL precipitation is also done for certain research purposes. Application of precipitation test, that it can be very sensitive in detecting antigen. It can detect antigen about 1 microgram of protein can be detected by this reaction. There are applications in forensic medicine, identification of blood and seminal stains are there, food adulteration is there, and there are various types of precipitation reactions which are not generally used nowadays. Are being tests like tube population test, where the tube population test is the Kahn test, immunodiffusion precipitation in gel, electroimmunodiffusions. So number of tests are there. We are going to briefly see a few of them. Ring test, simple is the precipitation and flocculation test is the ring test, which is simplest one, where you can add antigen solution over a column of antiserum in a narrow tube, and you can see the formation of precipitate form, where at the junction where the antigen and antibody flows towards each other, and where they meet and they combine, then they form the zone of equivalence, and those precipitation is formed. Example is Holt's thermoprecipitation test and Mansfield's grouping of streptococci. Then slide test includes a drop of, as we have discussed, a drop of antigen is there and antibody which are present in the serum. They are mixed and they are going to form the flocules, which are going to observed by flocules in the form of flocules, and this is VDR test for syphilis. Then tube test, that includes gun test again for syphilis. Then standardization of toxins and toxoids is there. And immunodiffusion in precipitation in gel, where the advantage of these tests that distinct band of precipitations are formed which are stable and can be stained for preservation. Number of different antigen in the reacting missions can be identified by this immunodiffusion test. So single diffusion in one dimension, which is routine procedure. Here you can see the antigen and antibody, they are mixed in one tube. Antigen is overlaid over the antibody or antiserum, and there is a band of precipitation in the meat in equivalence. Okay. The next example is Felix jet precipitation. This is done for corynebacterium diphtheria strain which are isolated, and they are sticked on a particular medium containing filter paper which is soaked with diphtheria antitoxin. If the strains are toxigenic, then they produce the toxin, and then this particular toxin, which is produced by toxigenic strains, are diffused in the medium, and they start diffusing in the medium, and the medium is already containing anti-toxin antibodies, and these antitoxin and toxin, where they meet, there is formation of arrow-shaped precipitation band. And these tests can also be used to know the relatedness between the two strains which are isolated during outbreaks. So this is the Felix jet precipitation, where you can see double diffusion in two dimension, which is a Opilonic procedure. Good example is the Felix jet precipitation test for corynebacterium diphtheria. It is most widely used test and helps to compare different antigen and antiserum which are identified. Next, we are not taking all the things, we are taking few examples. So agglutination reaction, where we are going to see the agglutination reaction, where this is a similar definition with that of precipitation, except what changes here is the antigen which takes part in the reaction is particulate antigen, and where the antigen antibody are going to form precipitate in the previous reaction. Here they are going to form the clumps or agglutinate, and therefore the antibodies which are agglutinating, here they are called agglutinates, more sensitive than the precipitation test for detecting the antibodies. There are certain incomplete antibodies which are monovalent antibodies, and they usually not take part in these reactions, or they are also known as blocking antibodies. Okay. Types of agglutination is direct, where the antiserum is mixed with the uniform suspension of antigen. So here also antigen antibody, they are in equal volume, they are mixed with each other, and there is formation of clump, and or agglutination is there, that can be seen. This is a example of slide agglutination test, which is grouping or identification of bacterial isolates can be there. In this case, what is important, agglutination reactions, what is important is to use the controls, which are positive control and negative control. And these tests can be read visibly, where precipitations require pediatric or previously they were used microscope. Nowadays RPR can be used, which can be showing the result with the naked eyes. Tube agglutination test example is the best example is the Vidal test, which is done for the identification and diagnosis of salmonella typhi, for the diagnosis of typhoid fever. The neutrophil agglutination test, Felix test for the diagnosis of rickettsial fever. These are the standard quantitative method for measurement of antibody, where serial dilution of the serum is done, and equal amount of antigen is added in each tube, and along with that controls are set up in that particular tube sets. Okay, so this is called as antigen tube agglutination reaction. Okay, so Felix is for rickettsial fever and Bidaritis is for diagnosis of typhoid fever. Here we can serially dilute the antibody suspension. One has to 20, one has to 40, one has to 80, one has to 120, one has to 160, like that, and then we can identify the baseline item as well as the significant item, as I have explained earlier. Okay, then hemagglutination, IBCs are used as antigen here. For example is the pulmonary test or cold agglutination test. So tube agglutination test can be seen as I explained to you. This is how we can do the serial dilution. When to take, that means the saline is taken in one tube, and in that tube, so you can see like this. If we are taking 1, 2, 3, 4, 5, 6, 7 tubes, so 7. Out of that, last one is the negative one. Okay. In each tube, we are taking a particular amount of saline. For example, 10 ml or 5 ml. Okay. In first tube, we are going to add. Before that, we are going to do the master dilution, where equal amount of saline and equal amount of serum is added, and that equal amount of saline suspension and serum suspension is transferred into this first tube with equal volume, suppose 5 ml. Okay. After mixing properly in the first tube, we are going to withdraw 5 ml into the second one, mix it properly, from there third one, mix it properly, remove, mix it properly, remove, mix it properly, remove, mix it properly, remove, and throw. So this is how we are going to do the serial dilution. Okay. And then we are going to add particular exact amount of antigen to each of the tube. Suppose 5 ml, 5 ml, 5 ml, 5 ml, 5 ml, 5 ml, 5 ml. Okay, in all of them. Okay. Then what will happen if the antibodies are present? Then they are going to show you, the presence of agglutination in the first tube, second tube, third tube, fourth tube, fifth tube, sixth tube. However, the number of antibodies are there which can occupy or react with the antigen which is added in that exact volume. They will show you the higher dilution. The last tube which will show you the agglutination, that will be considered as the antibody titer. Suppose here the last tube which shows agglutination is 1:40. After that, there is no agglutination. So 1, 2, 3 is the titer. 1:10, 1:20, and the last tube which shows the antibody presence of antibody which are agglutinating with the specific antigen is 1:40. So in this case, the titer is 1:40. Microscopic agglutination tests are performed in microtiter plate. Similar tests, similar procedures are followed, and the results are read under microscope. The example is the test which is done for the detection of leptospirosis infection. That is the MAT test, microscopic agglutination test. Okay. Then indirect or passive agglutination tests are there, where the soluble antigens are attached to the surface of particular carrier particles to convert precipitation reaction into agglutination reaction. So precipitation test into agglutination test. These are more convenient and sensitive. Here the carrier particles which are used are RBCs, latex particles or bentonite. Example of the test is ASO test. You can see this is the example. Drug coated latex particles are there. This L represents latex particle and these are the drug particles. Okay. So the drug is coated with this latex particle and antibodies. So antibody is having this antigen combined site. So wherever antibody is there, they can combine this. They can form a latex or lattice formation is there and agglutination is there. If we have this drug coated latex particle plus this antibody plus again only drug particles are there, and then we add the antibody, then drug particles, they can utilize these antibodies, these latex particles which are coated with the drugs, they will not be utilized here, so there will be no agglutination observed in this case. Okay? So passive agglutination tests, which are converting precipitation test to agglutination test, these are using chemically linked soluble antigens to inert particles such as latex or RBCs. So the soluble antigens, they are chemically linked to these latex particle or RBCs. In addition of a specific antibody will cause the particles to agglutinate. So we have the soluble antigen which is chemically linked to these latex particle. Now we are going to add specific antibody which is going to cause the particles to agglutinate. Example is the reverse passive agglutination test, where antibody are linked to a latex particle, is the Lansfield grouping instructed by. Another examples that can be used for detection of rheumatoid factors, anti-nuclear antibody in lupus erythematosus, antibody to base streptococcus antigens, and antibody to Trichinella spirans. Reverse passive agglutination, here instead of antigen, the antibodies are absorbed on the surface of carrier particles. These antibodies must be still reactive and is joined in such a way that the active sites are facing outwards. The antibodies are Y-shaped, so these antigen containing sites are facing outwards, and the antibody is absorbed to the latex particle at the bottom of the FC portion of the Y FC portion. This FC portion, here the latex particle is going to add. So latex particle coated to the known antibody plus serum or specimen which is added, in which the antibody we are looking, antigen we are looking for is there, and if the presence of specific antigen and antibody is there, then there is visible agglutination reaction. This is also sensitive, and the reason, the disadvantage is that this can give false positive results. Reverse passive agglutination can be explained with the help of principle. Here you can see these are the first antigen, the coated antibody on the RBCs, you can see here, and this is how they are going to form the complex, antigen antibody complex, and that is what the latex formation is there, and which is going to detect the antigen. And the example of the test which can be done are cholera toxin and detection of hepatitis surface antigen, reverse passive agglutination test. Then hemagglutination test as well, which refers to the agglutination test that are used using RBCs as a source of antigen. Various types of hemagglutination tests include direct hemagglutination and indirect hemagglutination. So direct hemagglutination, where the serum containing antibodies is going to be directly agglutinate with the surface antigen of RBCs to produce a map. Example is polymer test, polar agglutination, blood grouping, Coombs test, or anti-globulin test. Viral hemagglutination test, that in strict sense it is not an antigen antibody reaction. Here the hemagglutinin antigen which are present as a papillomas spikes or legends on the surface of some viruses, for example influenza viruses, right, which is having hemagglutinin and neuraminidase like spikes or papillomas or projections, and we are having the ability, or they are having the characteristic that we can agglutinate the RBCs which are having specific receptors on their substrate, the surface on this particular specific receptor. These hemagglutinin are going to attach, and they are going to aggregate the RBCs. They are going to clump the RBCs, and they are going to causing the hemagglutination. Now next test is the co-agglutination test, which is likely the latex agglutination test, agglutination techniques for detection of antigen as prescribed above. The protein A is a uniformly distributed cell wall component of particular strain of Staphylococcus aureus, which is known as O1 strain. And this O1 strain, this is having this particular protein A, and this protein A can bind to the FC portion. As I described you, antibody is Y-shaped, and and the below portion of this Y is the Fc region. Here this protein A binds, and these antibodies are maximum IgG antibodies to which this protein A are going to bind, and they are leaving the Fab region. That means the antigen combined sites free to interact with the antigen which are present in the suspension. Okay, and then we can see the visible agglutination of this Staphylococcus aureus particle that indicate antigen antibody reaction. That can be visible reaction. You can see in the picture we have the antibodies combined in the Fc region to this protein A of Staphylococcus aureus which is present in the cell component. These are the specific antigens and they are going to combine. This is going to prepare the latex and co-agglutination that can be seen in the larger form, or they can be observed properly. Next is well-Felix reaction, which is a heterophile tube agglutination test, where these tests where the patient's serum is tested for presence of agglutinins to co-antigen of certain non-motile strains of protease, which are OX19, OX2 and OXK. Now these protein and protease strains, various strains are there like OX19 and OX2, and the OXK strain of protease mirabilis. Now these strains, these antigens, they are sharing similarity with the antigens of rickettsia. Okay, so we cannot cultivate rickettsia in the lab. So for the identification of rickettsia fever, the heterophile antigens are utilized, and the test which is employed here is called as heterophile agglutination test. Here the antigens, they are sharing the antigenic property, the biological property and the chemical property, and therefore the antigen antibody, they are combined with each other. Another test is complement fixation test, which is rarely done nowadays, where you can see the ability of antigen complexes to fix the complement. Consists of 2 steps and 5 reagents. Example is Wassermann's reaction for syphilis. In this test, we can add an antigen in a tube. Again, we can add test serum to the specimen in which we want to identify the antibody against the specific antigen we are using and the complement. Okay. Now, if the antigen and antibody are going to combine here, they are going to form the antigen antibody complex, and the complement which we have added in the test is going to fix to this antigen antibody complex. So here the complement will be utilized, that will be fixed to the particular antigen antibody complex. Then we are going to add the hemolytic system. Now this hemolytic system is going to consist of RBCs as well as the ambocyte. Okay, but as the complement is utilized, which is already combined with the antigen and antibody, specific antigen, specific antibody complex, so there will be no hemolysis of this RBC and ambocyte combination, or hemolytic system is not utilized. So therefore, result will be no hemolysis. The test comes positive. Why? Because we have detected specific antibody in the patient's serum against specific antigen. But if negative test is there, what will happen? Antigen plus the test serum, which is having the antibodies but not against the specific antigen. Okay, so they are not going to form the antigen antibody complex. So here in this case, the complement is not going to be utilized. Okay, so it will remain free. It will not be fixed. It will remain free here. And then we are going to add this hemolytic system, which is having RBCs and ambocyte. Now this RBC and ambocyte means what? They are also having an antigen antibody complex, RBC and ambocyte. Either RBC acting as antigen, ambocyte acting as antibody. Their complex is there. So this complex, now this hemolytic system complex, this complement which is free is going to be added, and that is going to neutralize it. That is going to lyse the particular RBCs and ambocyte complex, and therefore there is hemolysis and the test will be negative because that is not identified properly. Here the whole system can be explained with the particular diagram. You can see the indicator system is there, the complement is there, which is added to the sheep red cells coated with the anti-sheep red cell antibody. Then complement reacts with the anti-sheep red cell antibody and lyes it. Okay, so this is the negative test. Where positive test, where we have the antigen, antibody, and the complement. Where antigen antibody is going to form the antigen antibody complex, where the complement is utilized, and then we are going to have the hemolytic system, which is not utilized. Complement will not be acting on them and not utilizing it, so it is no hemolysis over there, and the test is positive. Neutralization test is another test where the ability of the antibody to neutralize various effect of microorganism mediated through toxin, enzyme, and microbial antigens can be seen. Example is Naegele's reaction or Naegele's test, which is done for detection of the alpha toxin, which is produced by the Clostridium perfringens. Okay, where we are using a particular test. Medium, which is divided into two halves. On one half, we are going to strip the Clostridium perfringens strain. Another half also we are going to strip the Clostridium perfringens strain, and then we are going to overlay the antibody against the toxin, that is the alpha toxin. After doing this half, which is added with the toxin, is not going to show any type of presence of the opacity around the columns. Okay, this is because of the neutralization of the toxin. Where the half which is added with not added with the antitoxin, there you can see the opacity around the columns. So these are the expected questions from this particular topic. Enumerate the properties and types of antigen antibody reaction in detail. Describe in detail all the principal types and applications of precipitation reaction as well as agglutination. Short notes which can be asked on precipitation reactions, the types and application, agglutination reaction, the types and application, complement fixation test, neutralization test, methods of hemagglutination test, co-agglutination test. Okay, MCQs. Like prozoon phenomena is because of excessive antigen, excessive antibody, hyperimmune reactions, or both antigen and antibody excess. The example answer is B, excess of antibodies. Another MCQ, all agglutination reaction except Vidal, standard agglutination test, Vidal test, and Paul-Vanel test. Which is agglutination test? Paul-Vanel is the agglutination test. Standard tube agglutination test is there. Vidal is also agglutination test, but VDI is a precipitation test. So the answer is A, VDI, which is a precipitation population test. Thank you so much for your kind listening and observing all lecture, listening properly. Thank you so much. And I request you to kindly subscribe, like, and share our channel. Thank you so much. Have a good day. I hope you enjoyed this session. If you have any doubts, queries, you can ask in comment. Thank you so much.